Right, let's do this. Here we go. Welcome to this first me trying to do a lesson. I've got my t-shirt on, got my brew. So let's go for it. Right. First of all, I put this sheet onto Edmodo, and I shall put it on the email that I send you as well. And if you click on the link in Edmodo, it'll take you to this explain everything where I go through the labelling of this diagram. So that's a bit of revision. Let's play it a bit now.
Right, we're going to label up this summary diagram of transcription and translation.
My voice.
Here we go.
Oh.
I'm on new software here, so bear with me. Pen.
Pen. Amateur.
Should we go in blue? Right, we're going to do this in blue. Right. I'm going to label what I know first of all. Right, nuclear envelope. I can find that. That's easy. That's there. That's our nuclear envelope. Nuclear.
Right, enough of that. Yeah, so if you want to look at that, then please do. And just to have a bit more of a recap, we're also going to zoom through transcription, splicing, and translation. And we need to focus on keywords in biology, and therefore transcription. We need to talk about RNA polymerase, antisense strands of DNA. I think I can make this into a thing here. Antisense strand of DNA. Always names bonds. Name enzymes. Name another bond. Say what's made. Easy marks. Splicing. Again, introns removed, exons joined, and we make pre-mRNA into mRNA. And then translation, and these two words we must never confuse. That's where ribosomes are needed, etc. Anticodon, codon, hydrogen bonds, peptide bonds, ribosome moving. Again, it's all good marks, and I've summarised that on the diagram. Right, so we're just going to do some new stuff. We're going to look at transcription translation in eukaryotes and prokaryotes, and I've put some links to a couple of good videos that show this really well. First, it's just making sure that we know what eukaryotes are. Eukaryotes are cells, well, organisms that have cells with nuclei and membrane-bound organelles. So yeah, the animals do, the plants do, the fungi do, and the protists do, which is like our junk group where we put eukaryotes that don't fit into other categories, e.g. algae, amoeba in there as well. And then in the prokaryotes, we can just think of them as one thing. They're the bacteria. Where does transcription take place in eukaryotes? Well, that's inside the nucleus. And where does it take place in prokaryotes? Well, they don't have a nucleus, so therefore it must be somewhere in the cell, and all they have is cytoplasm. Is pre-mRNA produced in eukaryotes? Yes, it is. Now, is it made in prokaryotes? No, it's not. There'd be no point kind of going through this if there wasn't a major difference between them. Yeah, and that's the major difference. They don't make pre-mRNA. And because we do, as eukaryotes, we need to do splicing. Remove the introns, connect the exons to make mRNA from pre-mRNA. Prokaryotes, they don't make pre-mRNA. They go straight to mRNA. They don't have introns in their genes, and therefore it goes straight into mRNA. They don't need to do splicing. An extra step's missed out. Again, it makes them faster, they're simpler organisms. Then translation is carried out by, well, it's ribosomes and ribosomes, but how can we add a bit of extra A-level detail? Well, it's 80S ribosomes in eukaryotes, and it's 70S ribosomes in prokaryotes. We do need to be a little bit careful, because remember, in eukaryotic cells there are mitochondria, and some have chloroplasts. They have 70S ribosomes. Yeah, they have genes with no introns. Yeah, they are basically bacteria that have moved into a eukaryotic cell. Okay, so we need to know these extra little bits, and if you watch the video, that's what you should be doing. Making a table similar to this, add a bit more detail. That's all you really need to know for prokaryotes. What we're going to be moving on to, and the hard bit will come on our lesson on Friday, is looking at genetic diversity due to meiosis. So mutations we've done a bit on, about deletions, substitutions, etc. That's changing the genetic code. That's a mutation. A mutation is also a change in the amount of DNA in a cell, and that can occur when meiosis goes wrong. So this is just us recapping the difference between GCSE, mitosis and meiosis, and adding a bit more A-level detail. Again, I'd make your own copy of this table, pause it at the end, copy it out, maybe make the gaps bigger to add extra detail if you think it's needed from the spiel that I'm going through very, very badly. And a quick slurp of coffee. I called it a brew before, which might have led to the misconception it was tea. I do apologise. Anyway, mitosis. Again, this is GCSE knowledge. So mitosis, where does it take place in animals? The main mistake people make here is they say everywhere. Now, my brain cells are not doing mitosis, okay? So I need to be careful. And this is where I would say, the part of my body that is repairing itself. And the cells that do that are called stem cells. So the skin has a layer of cells. It's called the Malpighian layer. That's where the stem cells that are producing new skin cells. So not all stem cells differentiate and specialize. These are just cells that change into more skin cells. I could have said bone marrow there as well. Those are two good places, but if I go down the gym that I can't go to anymore, not that I did anyway, but if I did have my own gym and I started pumping iron, then I'd activate stem cells in my muscles and they'd start replicating by mitosis as well. So yeah, you need nuance in your answer. Where does meiosis take place? Again, the common error, people say sperm cells. Sperm cells make sperm cells. Well, they don't. Yeah, they are made in our sex organs. And the correct name, always gets a giggle, are gonads. Yeah, testes and ovaries. And again, it's not as straightforward because, males post-puberty are making sex cells all the time, whereas ovaries, they made sex cells when the individual was a fetus. So, once born, ovaries don't make sex cells, but they do kind of finish off the replication. So, again, tricky. It's why it's always easy just to say testes, because they are churning out sex cells. Next one, how many rounds of cell division? And we'll call it rounds, and we'll see why probably on Friday, but I'll allude to it now. Well, there's only one cytokinesis step in mitosis. At the end of the G2 phase, it goes through mitosis. So yeah, at the end, there's one. In meiosis, there's two. Obviously, when one replicates, we get two cells, and then there's another round, so we get four. Chromosomes are copied before cell division? Yes, they are. Are they meiosis? Yes, they are. Number of cells produced from one parent cell? Well, if there's one division, there's two. And if there's two divisions in meiosis, it's four. And cells are clones of parents in mitosis? Yes, they are. And hopefully you can write a nice big answer as to why, about identical chromatids separating and then the chromosomes being pulled to opposite poles. In meiosis, the whole point of it is no, they're not the clones. And therefore, why do we need mitosis? Well, as I said at the start, it's needed for, I can hear you all shouting out the answer, growth and repair of tissue. Whereas meiosis, it gives us variation, and variation is crucial for evolution. So an evolution just enables survival chance increasing in a species in a changing environment. So it allows evolution of species. Doesn't give an individual more chance because it depends on the environment. If you've got variation that gives you white fur, for instance, and you're living in a place where all the snow's gone now due to global warming, then you're not going to be well adapted. You'll be eaten by a hungry predator. You'll suffer what we call predation pressure. So in Friday's lesson, we're going to go through in detail how we get this variation in meiosis. And that's it from me for today. Hope you're all well, and I'll see you on Friday.